Welcome back to The Explainer. Okay, today we are tackling one of the absolute biggest mysteries in all of biology. Seriously, how does a single fertilized egg end up building something as insanely complex as a brain? Well, we're going to unpack the classic experiments that literally turned our understanding of early development completely upside down.
So I want you to imagine embryonic cells as basically completely blank slates. Right at the beginning, in those earliest stages, they have absolutely no identity. They're just waiting for instructions, a specific set of molecular signals to tell them what to become. And that sets up the central puzzle we're diving into today. How does a totally uncommitted cell figure out, hey, I'm supposed to differentiate into a brain?
For decades, scientists were hunting for these specific signals that trigger what we call neural induction. That's really just the fancy term for those initial steps where uncommitted embryonic cells start walking down the path to becoming part of the nervous system. And, the assumption for the longest time was that there simply must be some powerful active signal, like a brain-making chemical, that comes along and switches this pathway on. Makes sense, right?
Section 1. Meet the animal cap. To actually crack this code, developmental biologists turn to a totally classic model organism. We're talking about the amphibian embryo, and specifically, they focused on this one really fascinating region known as the animal cap. Take a look at the structure of this early xenopus, or frog, embryo. Up top, in the region marked as the ectoderm, you've got the animal pole. Now, in a normally developing embryo, this exact spot eventually turns into two very different things: ordinary skin, which we call the epidermis, and the entire neural tissue that will make up the nervous system.
So to figure out how the embryo makes this huge choice between skin and brain, scientists came up with the animal cap assay. And honestly, the steps are beautifully simple but incredibly powerful. First up, you physically explant, meaning you literally just snip out the animal cap ectoderm from the rest of the embryo. Next, you take that intact chunk of tissue and you culture it all by itself in a simple salt solution. And finally, step 3, you just wait and observe its final tissue fate. By taking it out, you are completely stripping away any instructive signals it might have been receiving from the rest of the embryo sitting below it.
Okay, so here is the baseline result, and it's absolutely crucial. When you just leave this isolated, intact animal cap to its own devices, it uniformly turns into epidermis, just ordinary skin. And honestly, this made perfect sense to early researchers. To them, it seemed like clear-cut proof that without some specific brain-inducing signal from the rest of the embryo, the tissue just kind of shrugs and defaults to becoming skin.
Section 2. The cell dissociation twist.
But then, scientists decided to tweak the assay, and this is where things get wild. You still explant the animal cap, just like before, but for step 2, instead of leaving it as this nice, solid chunk of tissue, you completely disperse the cells. You pull them all apart and put them in a medium that inhibits cell-to-cell communication. You are effectively diluting away any chemical whispers these cells might be sending to their immediate neighbors. Then, you just sit back and observe their fate.
Okay, let's dive into this, because the results here are absolutely mind-blowing. On the left, like we just saw, the intact animal cap becomes skin. But look at the right. The dissociated animal cap, those isolated, lonely cells that have been totally cut off from all communication with their neighbors, they spontaneously become neural tissue. They auto-neuralize. It turns out when you take away the signals, you don't get a blank slate at all. You get a brain cell. No way, right?
Section 3. The neural default model.
This quote right here perfectly captures just how profound this paradigm shift really is. Quote, cells of the embryonic ectoderm differentiate as neural tissue in the complete absence of signaling from their neighbors. Think about what that actually means. Cells do not need an active signal to become a brain. Neural tissue isn't some special VIP pathway they have to be shoved down. It is literally their default state. Of course, this revelation completely flips the script and forces us to ask the next logical question. If becoming a brain cell is the automatic, default program, then what on earth is telling that intact animal cap to become skin? There has to be some active signal that is constantly running interference and suppressing the brain program. Well, meet the culprits, BMPs, or bone morphogenetic proteins. I know, despite the name having bone in it, in early development, these TGF-beta signaling factors act as really potent neural inhibitors and epidermal inducers. So, in that intact animal cap, the cells are just constantly secreting BMPs, basically shouting at each other, "Become skin, do not become a brain." When the researchers pulled those cells apart in the dissociation assay, they diluted those BMPs, they lifted the suppression, which finally let that default neural program run free.
So let's put the molecular pieces together.
You have BMP, which is the neural inhibitor, and that leads to skin. But in a real living embryo, if you want to build a brain, a specific region called the organizer steps in. This organizer secretes BMP antagonists. These are cool little proteins with names like noggin and chordin. And their one and only job is to bind to BMP and block it. By blocking the inhibitor, they rescue that default neural state.
And this brilliantly illustrates the whole double negative logic of biology. Just look at those red T-shapes on the diagram. It's a system of inhibition. The organizer doesn't actually build the brain itself. It just secretes chordin, noggin, and follistatin to run interference. They block the BMP, which creates this protected safe zone in the ectoderm. And inside that safe zone, the tissue can finally realize its default destiny and develop into the head, the brain, and the spinal cord.
Section 4. From frogs to mammals.
Now, you might be sitting there wondering, okay, that's really cool for a frog, but what does that have to do with us? Well, the absolute beauty of developmental biology is conservation. When we look at mammalian neural specification, it's incredibly complex. The physical shapes and the timeline look totally different from our frog friend, but the fundamental molecular mechanisms are strikingly similar. A mouse doesn't have a frog's organizer, but it does have a tiny structure called the node. And that node performs the exact same protective, BMP-blocking function to kickstart the neural plate.
In fact, this whole strategy is universally conserved across vertebrates. Seriously, whether we're talking about the Spemen organizer in amphibians, the embryonic shield in fish, Henson's node in birds, or the node in mice, evolution basically stumbled upon this brilliant strategy, protecting cells from BMPs to unlock their neural default, and it just stuck with it across hundreds of millions of years of evolution.
And listen, this isn't just ancient evolutionary history. This has massive practical applications for us right now. Understanding this default model is exactly what allows scientists to work with embryonic stem cells in the lab today. By carefully controlling the environment in a petri dish and blocking those exact same BMP signals, we can actually coax human stem cells to follow their default path. We can prompt them to become specific therapeutic neural tissue.
Which leaves us with a truly exciting prospect. Think about it. The exact same molecular plot twist that a frog embryo uses to build a tadpole's brain is now sitting right in our hands. How might unlocking these ancient default programs totally revolutionize our approach to stem cell therapies? Could it hold the key to healing the human brain? It's wild to think it all started with just trying to understand isolated cells in an animal cap assay. Thanks for joining me on this explainer, and hey, always keep questioning the default.